For thousands of years, Tibetan communities have lived, worked, and raised children at altitudes that would leave most other human beings gasping for breath within minutes, routinely above 12,000 feet, in some cases well above 14,000, where the air holds barely 60% of the oxygen available at sea level. Scientists finally set out to understand exactly how that was biologically possible, expecting to find an unusually clever set of human adaptations built up gradually over many thousands of years of ordinary evolution. What they found instead traced back to a source nobody expected. A genetic gift passed down not from any human ancestor, but from an extinct species of archaic human that modern Tibetans' ancestors apparently met and interbred with tens of thousands of years ago. We're going to walk through exactly how researchers uncovered that connection, step by step, and by the end, you'll understand exactly why the origin of one of the most important genes in the entire Tibetan genome turned out to be one of the strangest and most compelling stories in all of human evolutionary genetics. Stay with me, because this one genuinely changes how you think about who modern humans have crossed paths with in our deep past. If ancient DNA, human evolution, and the real evidence-based story of our species' deep history interest you, this channel is for you. Hit subscribe, because we dig into discoveries exactly like this one every week. Let's start with the basic biological problem Tibetan populations have had to solve, because understanding just how serious that problem is makes the eventual solution so much more remarkable. At sea level, the air you breathe contains roughly 21% oxygen, and your body is finely tuned through hundreds of millions of years of evolution to function well at that specific concentration. As altitude increases, atmospheric pressure drops, and even though the percentage of oxygen in the air stays the same, the actual amount of oxygen available in each breath falls significantly. By the time you reach the altitudes where large Tibetan populations have lived continuously for thousands of years, the effective oxygen availability has dropped to a level that would cause severe altitude sickness and eventually life-threatening complications in almost any lowland-adapted human body without a lengthy, careful acclimatization process. To put that in perspective, the world's major mountaineering expeditions treat elevations above roughly 26,000 feet as what's informally called the death zone, where the human body, even an elite, fully acclimatized climbers, cannot sustain itself indefinitely and begins deteriorating regardless of fitness or preparation. Tibetan communities don't live anywhere near that extreme, but the towns, farms, and monasteries scattered across the Tibetan plateau routinely sit at elevations that would themselves qualify as extreme, dangerous altitude for an unacclimatized visitor arriving from sea level. And unlike a mountaineering expedition, this isn't a temporary, carefully managed physical challenge undertaken by trained specialists. It's the ordinary, everyday condition of raising children, farming, and living a full human life, generation after generation for thousands of years running. Most people who travel to high altitude, even after acclimatizing over days or weeks, still experience a stopgap biological response that works reasonably well short-term, but causes serious problems if sustained for a lifetime. The body responds to low oxygen by producing more red blood cells, thickening the blood to carry more oxygen-carrying hemoglobin per unit volume. That works as an emergency measure, allowing a temporary visitor to function reasonably well after a period of adjustment. But maintained permanently across an entire lifetime, that same response becomes dangerous. Chronically thickened blood raises the risk of blood clots, strokes, and a specific serious condition called chronic mountain sickness, marked by an excessive overproduction of red blood cells that actually makes the blood too thick to circulate efficiently, ironically making oxygen delivery worse rather than better over time, alongside significantly elevated risks during pregnancy and childbirth that would, left unaddressed by any further biological adaptation, place a real long-term limit on any population's ability to thrive at extreme altitude across many successive generations. This is exactly the problem that populations living permanently at high altitude around the world have had to solve in one way or another, and different high-altitude populations have arrived at genuinely different solutions. Andean populations in South America, who have also lived at extreme altitude for thousands of years, show elevated hemoglobin levels similar to the short-term lowlander response, but their bodies appear to have evolved additional adaptations that help manage the associated risks somewhat better than an unadapted lowlander's body would over a lifetime, including specific adjustments to how their hearts and blood vessels handle the resulting thicker blood over decades of exposure. Researchers studying East African high-altitude populations, particularly Finally, certain highland communities in Ethiopia have found yet a third distinct pattern again. These populations tend to show comparatively normal hemoglobin levels, similar to sea level populations, without the pronounced increase seen in Andean populations, achieved through a different set of physiological adjustments that researchers are still working to fully characterize at the genetic level. Interestingly, when researchers have looked for the specific Tibetan EPAS1 variant in these East African highland populations, they haven't found it, meaning this particular Ethiopian solution to chronic low oxygen survival appears to have evolved through an entirely separate genetic pathway of its own, involving different genes entirely, rather than representing some kind of shared or converging mechanism with either the Tibetan or the Indian case. That independence is itself scientifically informative. It tells researchers that evolution didn't have just one single toolkit available for solving this particular survival problem, and that geographically and genetically separate populations, faced with the same underlying physiological challenge, were each free to stumble onto whatever specific genetic solution happened to be available within their own ancestral gene pool at the time. 3 separate human populations, on 3 separate continents, facing essentially the identical underlying physiological challenge of chronic low oxygen exposure, arrived at 3 meaningfully different biological solutions. A striking real-world demonstration of how evolution doesn't necessarily converge on one single correct answer to a shared survival problem, especially when different populations start from different existing genetic backgrounds and face slightly different specific versions of the same broader challenge. These pattern evolutionary biologists refer to as convergent problem solving without convergent mechanism. The same broad outcome, survival life at extreme altitude, achieved through genuinely different underlying biology in each case, shaped as much by which genetic raw material each population's ancestors happened to already be carrying as by the shared environmental pressure itself. Tibetan populations, when researchers finally studied them in genetic detail, turned out to have found an entirely different and in some ways more elegant solution altogether compared to either of these other 2 high-altitude populations. Rather than dramatically increasing red blood cell production the way most other populations do in response to low oxygen, Tibetan populations living at extreme altitude show comparatively modest increases in hemoglobin levels, closer to what a sea level population would show, while still maintaining excellent oxygen delivery throughout the body. Their bodies appear to accomplish this through a different mechanism entirely, increasing breathing rate and adjusting blood vessel function to deliver available oxygen more efficiently, rather than relying primarily on thickening the blood itself. That distinction turns out to be medically crucial. It means Tibetan populations largely avoid the dangerous long-term consequences of chronic, extreme overproduction of red blood cells that affects unadapted individuals and even some other high-altitude populations living at comparable elevations. When researchers set out to find the specific genetic basis for this unusual and clearly effective adaptation, they focused on a gene called EPAS1, sometimes nicknamed in popular science writing as a kind of super-athlete gene, because of the central role it plays in regulating how the body senses and responds to low oxygen levels. EPAS1 acts as a master regulatory switch, controlling the activity of numerous other genes involved in oxygen transport and red blood cell production, essentially functioning as the body's central oxygen sensing thermostat. The gene works by producing a protein that becomes active specifically when cells detect low oxygen conditions, at which point it switches on a whole downstream cascade of other genes responsible for the body's various responses to that low oxygen state, including, but not limited to, the genes controlling red blood cell production. Because EPAS1 sits at that kind of central control point, a variant that changes how sensitively or how strongly this particular switch responds to low oxygen can end up reshaping the entire downstream physiological response, rather than affecting just one single narrow biological process in isolation. That's part of why a single gene variant here could have such an outsized, wide-ranging effect on Tibetan physiology. It wasn't tweaking one small parameter, it was recalibrating an entire coordinated response system near its very starting point. Genetic surveys comparing Tibetan populations against lowland East Asian populations, including Han Chinese populations that share a documented, relatively recent common ancestry with Tibetans, found something immediately striking. A specific variant of the EPAS1 gene associated with the more efficient, lower hemoglobin altitude adaptation seen in Tibetan populations showed up at extraordinarily high frequency in Tibetan populations, in some studies present in the large majority of individuals sampled, while showing up only rarely in a small single-digit percentage among lowland Han Chinese populations that Tibetans share their most recent common ancestry with. That kind of dramatic frequency difference between two closely related populations is exactly the signature evolutionary biologists look for when trying to identify genes that have been under strong recent natural selection, as opposed to genetic differences that simply accumulated gradually through ordinary genetic drift over time. Genetic drift alone, without selection actively favoring a particular variant, tends to produce comparatively modest, gradual frequency differences between closely related populations over the kind of timescale involved here. A difference this large and this specific, appearing in a single well-defined gene with a clear, testable biological function, directly relevant to the exact environmental challenge the population in question actually faces, is essentially a textbook signature of powerful, targeted natural selection at work. That's already a strong signal of intense natural selection having acted on this specific gene variant within Tibetan populations specifically, favoring it so strongly, so quickly, that researchers have identified it as one of the fastest cases of measurable genetic adaptation ever documented in human evolutionary history, estimated to have swept through the population in what is, in evolutionary terms, an extremely short window of time. By some estimates, potentially within just a few thousand years, a remarkably fast pace for a specific gene variant to go from rare to dominant across an entire population through natural selection alone. But the truly unexpected part of the discovery came when researchers tried to trace exactly where this specific, unusually beneficial version of the EPAS1 gene had originally come from. If Tibetan and Han Chinese populations share a relatively recent common ancestor, as other genetic evidence clearly indicates, the expectation would be that this beneficial variant either arose as a completely new mutation within the Tibetan population sometime after the two groups diverged, or was already present at low frequency in the shared ancestral population and simply became more common in Tibetans due to the strong selective advantage it provided at high altitude. Both of those explanations are, in principle, entirely ordinary, unremarkable evolutionary scenarios, exactly the kind of thing researchers would expect to find without needing any more exotic explanation. Neither of those straightforward explanations turned out to fit the actual genetic data. When researchers compared the specific sequence of this beneficial Tibetan EPAS1 variant against a broader set of reference genomes, including ancient genomes recovered from extinct archaic human populations, they found an unmistakable close match, not to any known modern human population, and not to Neanderthals, but to a Denisovan genome, sequenced from DNA extracted from a small bone fragment recovered from a cave in Siberia, representing an entirely separate, extinct branch of the broader human family tree that split off from the ancestors of modern humans and Neanderthals deep in our evolutionary past. This was, by every account from the researchers involved, a genuinely startling result when it first emerged from the data. Nobody had gone looking for a Denisovan connection specifically. The initial goal of the research was simply to identify which specific gene was responsible for Tibetan high-altitude adaptation and understand how strongly it had been selected for. The archaic genome comparison was, in a sense, a final confirmatory step, one somewhat as a matter of thoroughness once the unusual evolutionary signature of the gene had already been established through other methods. Finding a clear, strong Denisovan match at that stage transformed what had already been an interesting finding about human adaptation into a genuinely unexpected story about ancient interbreeding between separate human species. Think about what that comparison actually revealed. The specific genetic variant giving Tibetan populations their remarkable, life-saving high-altitude adaptation wasn't a fresh mutation that appeared within the human lineage after Tibetans and Han Chinese diverged from each other. It was inherited, largely intact from an entirely different, extinct human species, meaning that at some point in the deep past, ancestors of modern Tibetan populations interbred with Denisovans, and one specific gene variant passed down from that ancient encounter turned out tens of thousands of years later to be extraordinarily useful for an entirely different reason. Nobody involved in that original encounter could possibly have anticipated surviving permanently at extreme altitude on the Tibetan plateau. This finding connects to a broader and by now fairly well-established body of evidence showing that Denisovans and the ancestors of modern humans interbred on more than one occasion. As modern humans expanded across Asia tens of thousands of years ago, the same broader pattern responsible for the notably elevated Denisovan ancestry found in some Pacific and Southeast Asian populations that researchers have documented at other sites across the region. What makes that Tibetan EPAS-1 case so remarkable and so scientifically significant in its own right is that it represents one of the clearest, most rigorously documented examples anywhere in human genetics of archaic human DNA providing a specific, identifiable, and genuinely life-saving functional benefit to a living modern human population today, rather than simply persisting in the genome as a largely neutral genetic remnant of ancient interbreeding. It's worth understanding exactly how researchers were able to establish this connection with real scientific confidence, rather than it simply being a loose or speculative resemblance. When two populations share a genetic variant because they inherited it from a shared, relatively recent common ancestor, that shared variant is typically surrounded by other shared genetic material nearby on the same chromosome, reflecting the fact that DNA gets inherited in larger connected chunks. Researchers examined the genetic sequence surrounding the Tibetan EPAS-1 variant and found it carried a distinctive pattern of surrounding genetic material that matched the Denisovan genome specifically, in a way that would be extraordinarily unlikely to occur by chance alone or through any pathway, other than actually inherited inheritance from an ancestral population that had directly interbred with Denisovans at some point in the past. This kind of surrounding sequence analysis is what separates a genuinely rigorous genetic finding from a superficial coincidence. Two unrelated genetic sequences can occasionally resemble each other at a single specific point purely by chance, especially across a genome as large as the human genome, which contains over 3 billion individual base pairs. But an extended stretch of matching sequence spanning a large enough region and matching in enough specific detail is a different matter entirely. The statistical odds of that level of matching occurring by pure coincidence rather than through actual shared inheritance become vanishingly small, which is exactly the kind of statistical rigor that allowed researchers to publish this finding with genuine scientific confidence, rather than presenting it as a speculative possibility. Researchers also use the specific pattern and length of that shared genetic segment to estimate roughly how long ago the interbreeding event that introduced this variant into the ancestral human population likely occurred. Using the same basic logic, over many generations, recombination gradually breaks up any large inherited genetic segment into shorter and shorter pieces. So a longer, more intact shared segment generally indicates a more recent inheritance event, while a shorter, more fragmented one points further into the past. The estimates researchers derived placed this specific instance of interbreeding at a time consistent with the broader window during which modern humans are known to have overlapped and interbred with Denisovan populations across parts of Asia, tens of thousands of years before Tibetan populations themselves became genetically distinct from other East Asian populations. That timeline raises a genuinely fascinating implication worth sitting with. The specific Denisovan-derived EPAS-1 variant appears to have been carried at some background frequency within the broader ancestral East Asian population for a very long stretch of time, likely providing little to no specific advantage for populations living at normal, lower altitudes, where the variant's specific benefits around oxygen regulation simply weren't especially relevant to survival. It was only once a specific group of that broader ancestral population began settling permanently on the Tibetan plateau at extreme altitude that this previously unremarkable inherited variant suddenly became enormously advantageous, and natural selection, acting with unusual speed and force, rapidly increased its frequency within that specific population over a relatively short evolutionary timescale, while it remained comparatively rare in lowland populations, where it offered little particular benefit. There's a useful analogy for what happened here, even though no analogy captures a biological process perfectly. Imagine a specific tool sitting unused in the back of toolbox for generations, passed down without anyone quite knowing what it was originally designed for, because none of the ordinary household tasks anyone in the family regularly does ever call for it. Then, one branch of that family moves to a completely different kind of environment, faces a completely new kind of practical problem, and suddenly discovers that this specific, previously ignored tool happens to be exactly what's needed to solve it. While the rest of the family, this is a striking illustration of a broader principle in evolutionary biology that extends well beyond this one specific case. Genetic variation that appears neutral or even mildly disadvantageous in one environment can become dramatically valuable when a population's circumstances change, and that reservoir of existing genetic diversity, including diversity inherited from long extinct populations through ancient interbreeding, can end up being exactly what allows a population to adapt rapidly to a radically new environmental challenge, far faster than waiting for an entirely new beneficial mutation to arise from scratch would ever allow. Evolution, in a case like this one, didn't need to invent an entirely new solution to the problem of extreme altitude survival. It simply needed the right raw genetic material to already be sitting somewhere in the ancestral gene pool, waiting for the right environmental pressure to make it valuable. It's worth being precise about what this discovery does and doesn't tell us about the broader story of Tibetan population origins, because the EPAS-1 finding, while genuinely remarkable, represents just one specific gene out of the many thousands that make up the full picture of Tibetan ancestry. Broader genome-wide studies of Tibetan populations, looking well beyond this single altitude adaptation gene, show that the overwhelming majority of Tibetan ancestry traces back to the same broader East Asian ancestral population shared with Han Chinese and other neighboring populations, with genetic and archaeological evidence suggesting the ancestors of modern Tibetan populations settled the high plateau region and became genetically distinct from lowland East Asian populations, sometime within roughly the last 9,000 to 15,000 years, a period that saw a documented population expansion onto the plateau following the end of the last ice age, when climate conditions in the region became more consistently habitable for permanent human settlement. The archaeological and genetic evidence, developed through completely independent methods, converging on a broadly similar timeline, is exactly the kind of cross-confirmation that gives researchers real confidence in the overall picture. In other words, the Denisovan-derived EPAS-1 variant is not the origin of the Tibetan people as a population in any broad sense. It's one single, remarkably consequential thread woven into a much larger genetic story that is, in its overall shape, closely tied to the broader population history of East Asia. What makes this particular thread so significant is how disproportionately important it turned out to be for actually making permanent life at extreme altitude survivable and sustainable across many generations, allowing a population using largely the same broader East Asian genetic toolkit as their lowland neighbors to successfully occupy one of the most physiologically demanding environments any human population has ever permanently settled anywhere on Earth. There's also a valuable methodological lesson embedded in how this particular discovery unfolded, one that extends well beyond the Tibetan plateau specifically. Before ancient Denisovan and Neanderthal genomes were successfully sequenced, researchers studying any unusual, highly beneficial gene variant in a modern human population had no way to check whether that variant might trace back to interbreeding with an extinct, archaic human population rather than to an entirely modern human evolutionary origin. Now that reference genomes exist for both Denisovans and Neanderthals, researchers routinely check unusual or strongly selected modern human gene variants against those archaic reference genomes as a standard part of investigating their origin. And the Tibetan EPAS-1 case has become one of the field's clearest demonstrations of just how significant and how easily missed that kind of archaic genetic contribution can be without that specific comparison being made directly. Similar investigations, following the same basic method, have since identified other specific instances where a genetic material inherited from Neanderthals or Denisovans appears to provide identifiable modern benefits in specific modern human populations. Variants affecting immune system function inherited from Neanderthals show up at meaningfully elevated frequency in some modern populations. For example, apparently having provided a real survival advantage against certain pathogens that Neanderthal populations had already been exposed to for a long period before modern humans arrived in the same regions and interbred with them. Researchers have also identified Neanderthal-derived variants linked to skin and hair characteristics that appear to have offered adaptive value as modern humans first moved into the different light and climate conditions of higher latitude Eurasia, a region Neanderthal populations had already been successfully adapted to for a very long time before modern humans ever arrived there. The Tibetan altitude adaptation case remains one of the clearest and most thoroughly documented examples of this broader pattern, in part because the specific functional benefit involved, surviving permanently at extreme altitude, is so easy to directly measure and confirm through modern physiological research on living Tibetan populations today. Researchers can quite literally measure the blood oxygen levels, hemoglobin concentrations, and altitude sickness rates of people carrying versus not carrying this specific variant in real time in living communities on the Tibetan plateau right now, giving this particular case a level of direct, testable, real-world confirmation that many other proposed instances of archaic-derived adaptive benefit in populations where the specific advantage is harder to directly measure simply don't have available to the same degree. Put the whole picture together, and what emerges is a genuinely remarkable case study in how ancient encounters between different human species, tens of thousands of years in the past, can end up shaping the specific biological capabilities of a living human population today in ways nobody could possibly have predicted at the time the original encounter happened. Ancestors of modern Tibetan populations, at some point in the deep past, encountered and interbred with Denisovans, an entirely separate branch of the broader human family tree that has been extinct for tens of thousands of years. That encounter left behind a specific genetic variant that sat quietly within the ancestral population for a long stretch of time, providing little obvious benefit under ordinary lowland conditions, until a specific group of that population's descendants moved permanently onto one of the most oxygen-scarce inhabited environments on the entire planet, at which point that same ancient inherited variant became one of the single most important genetic factors, determining whether their descendants could survive and thrive there across dozens of subsequent generations. That's the real story behind a discovery that, on the surface, sounds like a simple question about why Tibetan populations handle high altitude so well. But underneath that simple question sits a genuinely profound story connecting the deepest layers of human evolutionary history to the everyday biology of millions of people living on the Tibetan plateau right now. An extinct human species that most people have never even heard of turns out to have handed down completely by accident one of the most consequential genetic gifts documented anywhere in modern human evolution. Not through any deliberate act, simply through the ordinary ancient process of two different human populations meeting, mixing, and each leaving a permanent trace inside the other, tens of thousands of years before anyone could have imagined how significant that specific trace would eventually become. Scattered throughout the it's remarkable in their day-to-day effect on how anyone actually lives. The Tibetan EPS1 story is a rare, clear window into what happens when one of those quiet, ordinary fragments turns out, under exactly the right circumstances, to matter more than almost anything else in the entire genome. A reminder that the deep past isn't sealed off from the present at all. It's woven directly into the biology of people living, breathing, and raising families at 14,000 feet right now, whether they've ever heard the word Denisovan or not. This changed how you think about human evolution, ancient interbreeding, or how much of who we are today traces back to encounters with species that no longer exist. Tell me in the comments what surprised you most. The speed of the genetic adaptation, the Denisovan connection itself, or how differently Tibetan and Andean populations solved the exact same survival problem. I read every comment, and it's usually where the next deep dive comes from. And if you want more of the real evidence-based story behind what your DNA actually reveals about our species' deep past, subscribe now. We're just getting started.